Welcome back to the deep dive. For years now, one of the really widespread and frankly debilitating aftershocks of the pandemic has been PSC, post-acute sequelae of COVID-19.
Yeah, absolutely.
And specifically, we're talking about that profound, persistent fatigue that just affects millions of people globally.
hmm It's not just feeling tired. It's deeper.
Exactly. It's this deep, life-altering depletion. And the really frustrating part, for the longest time, has been the lack of a clear mechanism. We haven't known why.
And, well, that lack of mechanistic clarity is exactly why the sources we're looking at today are so important. Really exciting, actually.
Okay.
We're diving into a pretty crucial piece of research.
Yeah.
It doesn't just, describe the symptoms. It actually identifies a highly specific, novel target.
A target, like for treatment.
Potentially, yes. Our mission today is really to understand this new mechanistic insight they've uncovered. Basically, the discovery of a specific inflammatory factor circulating in the blood that seems to directly link the immune system issues to, well, the failure of energy production right inside your muscles.
Okay, that sounds like a major step. And the specific factor, the big idea here, it centers on a protein called the Soluble IL-2 receptor, or SIL-2R.
That's the one, SIL-2R. The key finding, really, is that having elevated levels of this SIL-2R might be the direct cause of mitochondrial failure, the powerhouses in your skeletal muscle cells.
Wow. Okay, so if that holds up, we're shifting from talking about this sort of vague chronic condition to having a potential, very specific treatment target.
Exactly. It gives us a kind of roadmap. But to really appreciate the mechanism, I think we first need to understand the sheer physical problem these researchers actually observed in the patients.
Right, ground truth first. So to figure out this fatigue, you've got to start with the people actually experiencing it. Who were the participants they studied?
So they focused on a very specific group. It was 11 patients diagnosed with PSC. And critically, all of them had persistent fatigue for over 4 weeks after their infection.
Okay.
And importantly, these were all people living in the community. They hadn't been hospitalized. They'd only had mild to moderate initial COVID illness.
So this wasn't about, like, recovery from being on a ventilator or something.
No, exactly. This was aimed squarely at that long-term lingering fatigue that's affecting, so many people in the general population.
And the researchers, they were careful to match these patients. They had 17 healthy controls, and there were no significant differences beforehand, like in age or BMI, sex distribution.
Correct. Yeah. They wanted to rule out those general health differences as much as possible to really focus just on what the lingering effects of the virus itself might be causing.
Okay, so similar groups otherwise, but the functional difference you mentioned was pretty stark, right?
It really was. The PASC participants, they showed a statistically significant 16.4% lower average distance covered on a standardized 6-minute walk test.
16.4%. Almost a fifth less distance in just 6 minutes? That's a really tangible physical limitation.
It is, and that 16% drop, that's what translates into feeling like you just can't handle simple daily activities. You've got someone who looks physically healthy, but they simply cannot sustain effort. And here's where the functional puzzle, really came into focus. When the researchers actually measured things like muscle strength and muscle mass, everything looked completely normal in the PAC group.
Wait, hold on. So the physical structure, the engine itself was intact? The muscle fibers were there, the strength was okay, but the performance was way down.
Exactly. That's the paradox. It strongly suggested it wasn't just simple deconditioning, from being sick, or actual muscle wasting, atrophy.
So if the machinery itself is fine, the problem must be in how it gets its fuel or how it generates power.
Precisely. That led them directly to hypothesize there must be some kind of metabolic deficit, an energy problem.
Okay, so if the symptoms are pointing towards an energy problem deep inside the muscle, where did they look next to confirm that? They had to go inside, right?
They did. They performed muscle biopsies.
Right.
Specifically, they took samples from the vastus lateralis, that's the big muscle on the side of your thigh.
Your main walking and standing muscle, yeah?
Right, a powerhouse. And they were looking directly at the cellular energy factories, the mitochondria.
And what did they find inside? Did the cell machinery confirm the hypothesis?
It absolutely did. They used a technique called high-resolution respirometry. It basically measures how well the mitochondria are breathing, how much oxygen they consume to produce energy.
Okay.
And they found a dramatic reduction. About 30% lower mitochondrial oxygen consumption in the PASC patients compared to the healthy controls. The power output was just seriously crippled.
A 30% power cut. That's a massive deficit at the cellular level. And what's really fascinating, I thought, was that the sources detailed where exactly in the mitochondria the failure seemed to be happening.
Yes, and that specificity is such a critical clue. Mitochondria use this sequence of protein complexes, it's called the electron transport chain, to generate ATP, which is our cellular energy currency.
Right, the energy molecule.
And they found that the dysfunction was predominantly driven by a severe impairment in what's called complex I.
Complex I.
Think of complex I as maybe the crucial first step, like the ignition switch for the whole mitochondrial engine. If that switch is faulty, the entire energy production process just stalls out.
And they didn't just stop at measuring oxygen use, did they? They also checked, like, the supporting component.
That's right. They measured the activity levels of key enzymes, things like citrate synthase and cytochrome C oxidase, or CCO. These are kind of general markers for mitochondrial health and just overall quantity.
Right.
Both were significantly reduced. And furthermore, when they used Western blots to look at the actual amounts of the proteins that make up these complexes, they saw marked decreases across the board. Complexes 2, 3, and V were all down.
Okay, so less activity and fewer protein building blocks for the energy machinery. This brings up a really fundamental question then. Was the muscle failing to build enough new mitochondria, like a production or biogenesis problem? Or was something actively destroying the ones it already had?
And this, I think, is another aha moment in the research. They actually measured the rate at which the muscle cells were synthesizing new mitochondrial proteins, making the parts.
And what did they find?
Those synthesis rates were entirely normal.
Yeah.
The muscle cells were trying to build new power plants at a perfectly healthy rate.
Wait a minute. So the construction crew is working fine, building new parts, but the total number of functioning power plants is still way down.
Exactly.
That implies something else must be going on, like something external is actively tearing them down faster than they can be replaced, increased degradation.
Precisely. So now we've established this picture. A clear functional limitation in patients, a localized metabolic failure in the muscle centered on complex NIs, and now evidence suggesting this failure is likely due to some kind of active destructive signal.
But we still need the link back to the initial COVID infection and the immune system, right? What systemic factor is circulating around, traveling to the muscle, and basically telling it to break down its power supply?
That brings us to the next crucial part, the search for that systemic link. The researchers took blood serum from the patients and screened it for 65 different immune mediators, sort of casting a wide net.
Trying to find the messenger molecule delivering the bad news to the muscles?
And out of all those 65, one just shot way, way up, completely isolated from the rest. This soluble IL-2 receptor, SIL-2R, it emerged as the most dramatically elevated factor in the PASC patients by far.
And when we say most dramatically elevated, the sources mentioned concentrations up to 5 log-fold higher. Can you put that into perspective for us? What does a log scale like that mean?
Yeah, that's a great point because log scales can be confusing. It's not just 5 times higher. It's not even 10 times higher. A 5 log-fold difference means the concentration in these PAC patients was potentially tens of thousands of times higher than in the healthy controls.
Tens of thousands of times higher.
Yes. I mean, it's a signal so massive, so overwhelming, that the body simply couldn't ignore it.
And SIL-2R, that's known already, isn't it? It's an established marker of persistent T-cell activation.
That's right. It ties this finding directly back to the immune dysregulation that's known to be a characteristic of PSC. So it makes perfect biological sense that an immune factor like this would be the messenger carrying the signal.
But, okay, finding a correlation, even a massive one like that, isn't the same as proving causation, right? They needed to show that this SIL-2R itself actually causes the damage.
Exactly. Association isn't proof. They had to ask the critical question. If we take this SIL-2R factor at those high levels found in patients, and we put it directly onto healthy muscle cells in a lab dish, does it cause the same kind of mitochondrial damage we saw in the human biopsies?
And they did this experiment using C2C12 myotube cultures, which are basically muscle cells grown in a dish.
Yep, standard lab model for muscle cells. They treated these myotubes with SIL-2R at concentrations mimicking what they found in the PAC patient's blood.
And the result?
It was striking validation. The SIL-2R treatment, all by itself, caused the energy production, the respiration rates, to just plummet. Basal respiration, maximal capacity, the amount linked directly to making ATP. It all fell significantly.
So just adding this one factor choked off the energy supply in the lab cells.
It did. And here's maybe the most crucial detail, the one that really nails the mechanism. They also tried treating the muscle cells with IL-2 alone. That's the main signaling molecule, the cytokine, that this receptor usually binds to in the body.
What happened then?
Nothing. IL-2 alone had no effect on the muscle cell respiration.
Ah, okay. So that proves it's the soluble receptor fragment itself, the SIL2R, that's the bad actor here. It's acting pathogenically on its own, independent of the usual IL-2 signaling pathway.
Exactly. The very molecule that gets shed as a marker of persistent immune activation seems to be the direct toxin causing organ-specific damage.
And just to tie it all together, did that in vitro SIL2R treatment also affect the mitochondrial protein levels in the dish, like they saw in the patients?
It did. It specifically reduced mitochondrial complex 3 protein levels in those lab-grown muscle cells, which perfectly mirrored the damage patterns they observed in the actual muscle biopsies from the PSC patients.
Wow. So the chain of evidence seems incredibly strong. You find this factor massively elevated systemically. You show that factor alone causes the specific local damage seen in patients.
Right. The proposed mechanism becomes pretty clear. Persistent immune activation after the initial infection leads to this huge surge in SIL-2R. This SIL-2R travels through the bloodstream, gets to the muscles, and somehow binds to receptors on the muscle cell surface. They suggested potential candidates like beta-1 integrins, maybe others.
And that binding translates the systemic immune problem into specific mitochondrial dysfunction within the muscle tissue itself.
That's the idea. Systemic inflammation causing local organ damage via this specific mediator.
This really shifts the whole perspective then, doesn't it? It moves us away from just describing symptoms and towards thinking about actual intervention. If SIL2R is the main circulating culprit, what does that mean for treatment possibilities?
Well, this is the so what part that truly matters for patients, I think. For many chronic fatigue syndromes, doctors often have to resort to pretty broad immunosuppression.
Right, things that dampen the whole immune system, which carries its own risks.
Exactly. You risk making people vulnerable to infections and other issues. But targeting SIL2R specifically offers a much more, surgical approach.
Like a targeted strike instead of carpet bombing the immune system.
Precisely. You're aiming to neutralize the specific pathogenic inflammation, the damaging part, while ideally leaving the beneficial parts of the immune response intact.
So what specific approaches did the researchers suggest could actually target or neutralize this SL2R?
They laid out a few potential translational pathways. The most direct would be developing, say, neutralizing antibodies that specifically bind to and inactivate SIL2R, or perhaps small molecule receptor antagonists.
Okay, drugs designed specifically for this target.
Right. But they also noted that some existing therapies might actually work through this mechanism, even if we didn't realize it before. For example, intravenous immunoglobulin, IVA. It's known that IVA treatments can sometimes lower SIL2R levels in the blood.
Ah, so that might give clinicians some immediate avenues to explore using therapies already available.
Potentially, yes.
Yeah.
It needs more study, of course, but it's an intriguing possibility.
And thinking beyond just active treatment, SIL2R could offer another crucial benefit that's often missing in these kinds of chronic, hard-to-pin-down illnesses, a reliable biomarker.
Absolutely. That's a huge point. If this connection holds up, measuring SIL2R levels in the blood could become a really valuable tool.
How so?
Well, it could serve as a quantifiable measure, something objective to track how severe a patient's PSEA is. And crucially, you could use it to monitor how well they're actually responding to any targeted treatments aimed at lowering SIL2R or blocking its effects.
So you move from relying solely on subjective symptom reports, how tired you feel today, to having concrete, objective biochemical data to guide treatment.
Exactly. That's a big step forward for managing chronic illness.
And the significance here, it feels like it extends much further than just COVID-19, right? We know that chronic fatigue syndrome, MECFS, has affected people for decades, often kicking off after other viral infections.
That's a very important connection to make. If this SIL2R mitochondrial dysfunction axis is confirmed and holds true more broadly, it could potentially provide the first concrete, unified mechanism underlying many of these post-viral fatigue syndromes.
So this one study focused on PSS might actually help unlock mysteries that have plagued patients with MECFS and similar conditions for decades.
It really could. It has the potential to revolutionize not just how we treat PSC, but how we understand and approach chronic fatigue syndromes across the board. It's quite profound.
Now, before we wrap up, we always need to acknowledge that science is an ongoing process. What limitations did the authors themselves point out in their research letter?
Yeah, they were very clear and upfront about that. The primary limitation they noted was the small sample size. It was only 11 PSC patients.
Which means you have to be a bit cautious about generalizing the findings too broadly.
Right. Although it's worth saying, the consistency and the sheer size of the effects they observed across multiple different types of measurements, functional, biochemical, protein levels, does give the data significant weight, despite the small n.
And they also mentioned the lab model, the C2C12 muscle cells.
Yes, they pointed out that while those cell lines are useful tools, they aren't a perfect replica of mature human muscle tissue. They suggested that follow-up studies using primary human muscle cells, cells taken directly from people, would be a critical next step for confirmation.
Okay, so important caveats, but the core finding seems robust.
I'd say so. Overall, the central finding really stands out. Highly elevated SIL-2R appears to be a specific circulating factor that mediates, or at least strongly contributes to, the muscle mitochondrial dysfunction driving post-COVID fatigue. It feels like a genuine breakthrough.
It really does. This research provides perhaps the strongest evidence we've seen yet for a clear biological mechanism and, importantly, a path towards specific evidence-based treatments for potentially millions of people.
So wrapping this up and thinking about where we go next, given the really compelling strength of this data pointing towards SIL-2R as the key mediator, here's something provocative for you, the listener, to consider.
Okay.
We now seem to know the what SIL-2R and the where muscle mitochondria. But the precise how still remains the ultimate molecular challenge, doesn't it? The authors themselves noted that the exact molecular mechanisms of how SIL-2R actually exerts its effect, specifically how it physically binds to the muscle cell surface. And is this dramatic sensitivity unique only to muscle tissue, or do other tissues get affected? These questions still require intense investigation.
Understanding that exact binding interaction.
Exactly. If researchers can definitively lock down that specific receptor SIL-2R is binding to on the muscle cell and understand that downstream signaling, well, then the pathway to designing a highly targeted effective cure becomes much, much shorter.
That molecular mechanism, that's the next deep dive for the researchers, and hopefully something we can revisit here soon. That looks like all the time we have for today. Thank you for diving deep with us.
Always a pleasure.